Welcome to the deep dive. Today, we're really getting into the weeds on a fundamental process in your body's energy game, gluconeogenesis.
Mm-hmm.
Think of it as your internal glucose factory, right? Building this fuel from things that aren't carbs. We've gathered a lot of detailed info, and honestly, our goal today is mastery. We want to understand every single aspect.
Exactly. Gluconeogenesis, it's your body's really clever strategy to keep glucose levels steady,
Right.
especially when you're low on dietary carbs, maybe fasting or something.
Yeah.
And this is absolutely vital. Some tissues, especially your brain, they really depend heavily on glucose. So even if you're not eating sugar, your body has this sophisticated backup. It's essential.
Okay, so if it's not carbs, where are the building blocks coming from? What's the raw material for this glucose factor?
Good question. The main players are glucogenic amino acids. These come from breaking down proteins in your body.
Okay.
Then you've got lactate, this stuff your muscles produce during hard exercise, and red blood cells make it too. Glycerol is another one. It's the backbone molecule of fats, triglycerides.
Right, from fat breakdown.
Exactly. And finally, there's propionate. It's a short-chain fatty acid. Plays a much bigger role in, say, cows, but we use it a bit too.
Interesting. And where does all this biochemical magic happen? Where's the glucose actually getting made?
Well, the liver is definitely the main hub, the undisputed champion, you could say.
Okay.
But the kidney is also a major player, particularly when fasting gets prolonged. After a few days without food, the kidney can actually become a significant source of glucose going into your blood.
Oh, okay.
And interestingly, the cells lining your small intestine, they have the enzymes too, and they can use propionate. But during fasting, the intestine itself is using glucose, so it makes from propionate probably just gets used right there locally.
Got it. So it's not just the chemistry, it's also about location and logistics. Now, what controls how fast this factory runs? What determines the rate the liver makes this new glucose?
Right. There are basically four key factors controlling the speed. First, just having the raw materials available, those substrates we just talked about.
Makes sense. Supply.
Second, the liver's ability to actually grab those substrates from the blood, uptake capacity.
Okay.
Third, the amount and the activity level of the specific enzymes that do the work, the ones in the gluconeogenic pathway itself.
The machinery.
Exactly. And finally, the liver's overall energy situation. Its oxidative capacity needs energy mostly from burning fat to power this whole process because making glucose costs energy.
Right.
And it also needs energy to handle the waste, like the nitrogen from amino acids, getting rid of it as urea.
Okay, so it's a balancing act. You need the materials, a working liver, the right enzyme tools, and enough power to run everything. Now, the name itself, gluconeogenesis, neo meaning new, it implies making something new, not just reversing things. Why can't the body just run glycolysis, the glucose breakdown pathway, backwards?
That's a really critical point.
Yeah.
Gluconeogenesis isn't just glycolysis in reverse. And the reason is that three specific steps in glycolysis are, well, effectively irreversible under normal cell conditions. These are the reactions catalyzed by hexokinase or glucokinase in the liver,
Right.
phosphofructokinase, which is a major control point, and pyruvate kinase, the last step. So to make glucose, the cell has to find ways around these roadblocks. It uses different enzymes, different reactions, clever detours.
Okay, let's map out these detours. How does it get past that pyruvate kinase step, the one that makes pyruvate at the end of glycolysis?
Right, this bypass is actually a two-stage affair happening in different parts of the cell. First, inside the mitochondria.
Okay, inside.
An enzyme called pyruvate carboxylase takes pyruvate. It needs ATP for energy and also the vitamin biotin as the cofactor.
Biotin, right.
It adds a carbon dioxide molecule, turning pyruvate into oxaloacetate.
Oxaloacetate.
Okay. Now, oxaloacetate itself can't easily get out of the mitochondria, so it gets temporarily converted into malate. Malate can be transported out into the main cell fluid, the cytosol.
Ah, the shuttle system.
Exactly. Once malate is in the cytosol, it's converted back to oxaloacetate. Then the second enzyme of this bypass, phosphoenolpyruvate carboxykinase, usually called PBCK, takes over.
PPCK.
Got it. PPCK uses GTP, another energy molecule, not ATP this time.
Interesting, GTP.
It removes that CO2 that we just added and slaps on a phosphate group, and boom, oxaloacetate becomes phosphoenolpyruvate, or PEP, which is back on the main gluconeogenesis highway.
Okay, wait, GTP, that's unusual. Usually it's ATP. Where does the GTP come from, specifically in the liver and kidney doing this?
Good catch. In the liver and kidney, there's a specific reaction in the citric acid cycle, the one catalyzed by succinate thiokinase, that actually produces GTP directly, instead of ATP like in most other tissues.
Ah.
And this is metabolically really elegant. It directly couples the activity of the citric acid cycle, the main energy hub, to the process of making glucose.
Why is that link so important?
It helps prevent the cell from draining too much oxaloacetate away from the citric acid cycle just to make glucose. If you pull too much oxaloacetate out, the cycle itself can stall.
Right. It needs oxaloacetate to keep running.
Exactly. So you need this balance between anaplerosis filling up the cycle intermediates and cataplerosis removing them for other things like gluconeogenesis here. Using GTP from the cycle helps maintain that crucial balance.
That makes perfect sense. A really tight regulatory connection. Okay, detour 1 navigated. How does the cell bypass phosphofructokinase, or PFK, that key enzyme that commits glucose down the glycolysis path?
This bypass is a bit more straightforward. There's an enzyme called fructose 1-6-bisphosphatase.
Okay.
It basically just does the opposite of PFK. It removes a phosphate group from fructose 1-6-bisphosphate, converting it back to fructose 6-phosphate.
Simple enough. Simple enough reaction-wise, but where this enzyme is located is significant. You find it in the liver, kidney, and also skeletal muscle. That fits with their roles in either making glucose or making glycogen from smaller bits.
Right.
But it seems to be absent, or at very low levels, in heart muscle and smooth muscle.
What does that imply?
It suggests those tissues probably handle glycerol, another gluconeogenesis substrate, a bit differently. They might use it more for their own energy needs rather than contributing significantly to making glucose for the whole body via this specific bypass.
Interesting tissue specificity.
Okay.
Last bypass.
Yeah.
How do we undo the very first step of glycolysis, the hexokinase or glucokinase reaction that traps glucose in the cell by phosphorylating it?
For this final hurdle, we use the enzyme glucose 6-phosphatase.
Okay.
Its job is simply to remove that phosphate group from glucose 6-phosphate, releasing free glucose.
And that glucose can leave the cell.
Precisely. And crucially, this enzyme, glucose 6-phosphatase, is really only found in the liver and the kidney cortex, the outer layer of the kidney. It's notably absent in muscle. Ah. And that's why muscle can't release glucose.
That's exactly why. Muscle can store glucose as glycogen, absolutely. But when it breaks down that glycogen, the glucose 6-phosphate produced is stuck inside the muscle cell. It has to be used for the muscle's own energy needs. It can't be exported to raise blood sugar.
So the liver and kidney are the glucose exporters, thanks to glucose 6-phosphatase. Okay, we've covered pyruvate, glycerol entering. How do the other substrates feed in, like amino acids and lactate?
Right. So glucogenic amino acids, meaning the ones that can be turned into glucose, they first need to get rid of their nitrogen group through processes like transamination or deamination.
Okay.
What's left, the carbon skeleton, is then converted into either pyruvate itself or into one of the intermediates of the citric acid cycle, like alpha-ketoglutarate or oxaloacetate.
So they can jump into the pathway at different points.
Exactly. It connects protein breakdown directly to carbohydrate synthesis.
Yeah.
And lactate. Well, lactate can be converted back to pyruvate by an enzyme called lactate dehydrogenase. So lactate is basically just another way to supply pyruvate for gluconeogenesis. It's all interconnected.
Makes sense. Now, what about propionate? You mentioned it earlier, especially in ruminants. How does that fit in?
Yeah, propionate is a really big deal for animals like cows and sheep. Their digestion involves a lot of fermentation by gut microbes, producing huge amounts of these short-chain fatty acids, including propionate.
So it's a major fuel source for them.
A major glucose source, actually. It contributes significantly to their blood glucose. The pathway to get it into gluconeogenesis is a bit involved. First, propionate gets activated with coenzyme A to form propionyl-CoA.
Okay.
Then an enzyme called propionyl-CoA carboxylase, another one that needs biotin, adds a CO2, making D-methylmalonyl-CoA.
Another carboxylation.
Yep. Then a race-MACE enzyme flips it to the L form, L-methylmalonyl-CoA. And finally, the key step, methylmalonyl-CoA mutase, which needs vitamin B12 as a cofactor, rearranges it into succinyl-CoA.
Succinyl-CoA, that's a citric acid cycle intermediate.
Bingo. So propionate gets converted into something that can feed right into the central metabolic hub, and from there, oxaloacetate can be generated for gluconeogenesis.
And you mentioned vitamin B12 dependence. Is that clinically relevant for us?
Absolutely. If someone is deficient in vitamin B12, this methylmalonyl-CoA mutase enzyme doesn't work properly.
Right.
This causes methylmalonic acid to build up and get excreted in the urine. It's a condition called methylmalonic aciduria, a classic sign of B12 deficiency.
Okay. So for humans, propionate isn't a major source.
Not major, no. We get small amounts from breaking down odd-chain fatty acids found in some fats, especially dairy and meat from ruminants, also from metabolizing the amino acid isoleucine, and a tiny bit from cholesterol breakdown. It contributes, but it's not like in cows.
Got it. Specialized player for us. Okay, last substrate, glycerol, from fat breakdown.
Right. Glycerol is released from our fat stores, adipose tissue, when triglycerides are broken down. That's lipolysis.
Mm-hmm.
In the fed state, the liver might use some of that glycerol to remake triglycerides, but it's also a direct substrate for gluconeogenesis. And during fasting, when lipolysis really ramps up to provide energy, glycerol becomes a really significant source of carbon for making new glucose, both in the liver and the kidneys. It enters the pathway pretty easily. It gets phosphorylated to glycerol 3-phosphate, then converted to dihydroxyacetone phosphate, or DHAP.
DHAP, isn't that also in glycolysis?
Exactly. It's an intermediate that sits right at the crossroads of glycolysis and gluconeogenesis, so glycerol slots in very neatly.
Okay, we've mapped the pathway, the detours, the entry points. Fantastic detail. Now, the big question. How does the body stop glycolysis, breaking down glucose and gluconeogenesis, building it, from just running at the same time and wasting massive amounts of energy?
Yes, the crux of the regulation. This is where the concept of reciprocal regulation is absolutely essential.
Reciprocal regulation.
Yeah. It's all about preventing what's called a futile cycle, pointless energy expenditure, by having opposing pathways run full tilt simultaneously. This tight, coordinated control gives your body incredible metabolic flexibility.
So it can switch gears smoothly.
Precisely. Switch between using glucose when it's abundant and making glucose when it's scarce, depending on, what you've eaten, your energy demands, all that.
What are the main strategies the body uses for this regulatory dance?
There are basically 3 main levels of control acting here. First, and slowest, is changing the amount of the key enzymes. This involves controlling gene expression, turning genes on or off, induction and repression. This takes hours to really take effect.
Okay, long-term adaptation.
Exactly. So when glucose is high and insulin is around, the genes for glycolysis and fat synthesis enzymes get ramped up, while gluconeogenesis enzyme genes get turned down. When glucose is low and hormones like glucagon and cortisol are dominant, it's the reverse.
Makes sense. What's the second level?
Second is covalent modification. Usually this means adding or removing phosphate groups from existing enzyme proteins. Reversible phosphorylation. This is much faster minutes.
Like flicking a switch on the enzyme?
Kind of, yeah. Hormones like glucagon and epinephrine, which signal low glucose or stress, trigger a rise in cyclic AMP, or CANAMP.
The second messenger.
Right. c-MeK activates protein kinase A, or PKA. PKA then phosphorylates key enzymes. For example, it phosphorylates pyruvate kinase, which inactivates it, slowing glycolysis down.
Okay.
This phosphorylation simultaneously favors gluconeogenesis. Glucagon also does something else. It boosts the transport of amino acids into the liver, making sure the raw materials are there for glucose synthesis.
Clever. And the third level, the fastest.
That's allosteric regulation. This is instantaneous. Small molecules bind to enzymes at sites other than the active site, and this binding immediately changes the enzyme's activity, up or down.
Allosteric. Like dimmer switches controlled by metabolic signals. Give us some key examples here.
Okay, a great one for gluconeogenesis is pyruvate carboxylase, that first bypass enzyme in the mitochondria. It's strongly activated by acetyl-CoA.
Acetyl-CoA from fat breakdown.
Primarily, yes, especially during fasting when you're burning fat for energy. So when fat oxidation is high, you make lots of acetyl-CoA. This acetyl-CoA does 2 things. It provides fuel for the citric acid cycle to make ATP.
Right.
And it directly signals pyruvate carboxylase to ramp up glucose production. It essentially says, we have plenty of fuel from fat, let's make glucose.
And does it affect pyruvate going the other way?
It does. Acetyl-CoA also inhibits pyruvate dehydrogenase, the enzyme complex that would normally convert pyruvate to acetyl-CoA to be burned in the citric acid cycle.
So it diverts pyruvate towards glucose synthesis.
Exactly. It ensures pyruvate is used for making glucose, not for being oxidized when fat is the main fuel. Plus, the high rate of fat oxidation itself provides the huge amount of ATP needed to actually power gluconeogenesis. It's beautifully integrated.
That really is elegant. What about allosteric controls hitting glycolysis, stopping glucose breakdown?
Right. Phosphofructokinase 1, that key glycolytic enzyme, is inhibited by high levels of ATP. Makes sense if the cell has lots of energy, slow down the pathway that makes more. Sure. It's also inhibited by citrate, an early intermediate in the citric acid cycle. High citrate also signals energy abundance and plenty of building blocks.
So energy-rich signals turn glycolysis down. What turns it up?
The big activator is AMP adenosine monophosphate. AMP levels rise sharply when ATP is being used up. Think of AMP as a really sensitive fuel gauge for the cell.
More sensitive than ATP itself.
Much more. A small drop in ATP can cause a much larger percentage increase in AMP because of an enzyme called adenylate kinase that balances ATP, ADP, and AMP. So when AMP levels climb, it powerfully activates PFK1, screaming, "We need more ATP! Run glycolysis!"
Wow.
And there's a knock-on effect. When ATP inhibits PFK1, its substrate, fructose 6-phosphate, builds up. This buildup, in turn, inhibits hexokinase in most tissues.
The first step of glycolysis.
Right. Slows down glucose uptake. But remember, glucokinase in the liver. It's not inhibited by glucose 6-phosphate. So the liver can keep taking out glucose, even when PFK1 might be slowing down a bit, allowing it to store glycogen.
Okay, so cell energy status is a huge controller via ATP and AMP. Now, you mentioned this one molecule earlier, fructose 2,6-bisphosphate. Sounds complicated, but you said it's really central, especially in the liver. What's its story?
Ah, yes. Fructose 2,6-bisphosphate. This molecule is, well, kind of the master switch in the liver for controlling glycolysis versus gluconeogenesis. It's incredibly potent.
How so? What does it do?
Okay, two main things. First, it is the most powerful known activator of phosphofructokinase 1, PFK1, the glycolytic enzyme. It basically overrides the inhibition by ATP and makes PFK1 much more effective, strongly stimulating glycolysis.
Okay, pushes glucose breakdown.
Exactly. And second, it's a strong inhibitor of fructose 1,6-bisphosphatase, the enzyme that does the opposite step in gluconeogenesis.
So it blocks glucose synthesis at that step.
Precisely. It increases the CMAP of the bisphosphatase for its substrate, making it less efficient. So high levels of fructose 2,6-bisphosphate mean go glycolysis, stop gluconeogenesis. Low levels mean the opposite.
And what controls the level of this master switch molecule itself?
That's the really clever part.
Right.
Its concentration is controlled by both substrate availability, basically how much glucose is around, and by hormonal signals. It integrates both types of information.
How is it actually made and broken down?
It's synthesized from fructose 6-phosphate by an enzyme called phosphofructokinase 2, or PFK2. Now, here's the twist. The same protein that has PFK2 activity also has the activity that breaks down fructose 2,6-bisphosphate. That activity is called fructose 2,6-bisphosphatase, or FBPase2.
Wait, this same enzyme does both opposite things. How does that even work?
It's a bifunctional enzyme, two different active sites on one polypeptide chain. It's genius, really.
Okay, so how is that bifunctional enzyme controlled? How does it know whether to make or break fructose 2,6-bisphosphate?
Dual control again, allosteric and hormonal. First, allosteric. Fructose 6-phosphate itself, the precursor. When glucose levels are high, fructose 6-phosphate levels rise. This stimulates the PFK2 kinase activity and simultaneously inhibits the FBPase2 phosphatase activity of the bifunctional enzyme.
So high glucose, more fructose 6-phosphate, more fructose 2,6-bisphosphate, more glycolysis.
You got it. Now, hormonal control. This comes mainly from glucagon, the hormone signaling low blood sugar.
Right.
Glucagon raises CAMP levels, activating protein kinase A, pKa, as we discussed. pKa then phosphorylates the bifunctional enzyme.
Okay, adds a phosphate. What does that do?
Phosphorylation inactivates the PFK2 kinase part and activates the FBPase2 phosphatase part.
Ah, so glucagon causes the breakdown of fructose 206-bisphosphate.
Exactly. Fructose 206-bisphosphate levels drop. This removes the strong activator of glycolysis, PFK1, and removes the inhibitor of gluconeogenesis, FDPase1. The net result: gluconeogenesis speeds up, glycolysis slows down. It's an incredibly sensitive and effective switch controlled by hormones.
That is truly elegant molecular engineering. Wow. Okay, so fructose 2,6-bisphosphate is the conductor. You also briefly mentioned xylulose 5-phosphate playing a role. How does that fit in?
Yeah, that's another layer linking to a different pathway. Xylulose 5-phosphate is an intermediate in the pentose phosphate pathway, the pathway that makes NADPH for synthesis reactions and ribose sugars for nucleotides.
Okay.
When glucose levels are high, more glucose also flows to the pentose phosphate pathway, so xylulose 5-phosphate levels rise. It turns out that xylulose 5-phosphate can activate a different enzyme, a protein phosphatase.
Another phosphatase.
Yes, this one removes phosphate groups from the bifunctional enzyme, but likely at a different site than the one pKa adds phosphate to. This dephosphorylation tends to activate the PFK2 kinase activity.
So high glucose, more pentose phosphate pathway, more xylulose 5-phosphate activates PFK2, more fructose, 2-picol-6-bisphosphate, more glycolysis.
Exactly. It provides another feed-forward loop. It links high glucose availability not just to glycolysis, but also indirectly to things like fatty acid synthesis, because the pentose phosphate pathway makes the NADPH needed for that too.
Wow, layers upon layers of regulation. Okay, we've hit enzyme amounts, phosphorylation, allosterics, this fructose 2.6-bisphosphate system. What about those futile cycles or substrate cycles you hinted at earlier? If regulation is so tight, why allow any simultaneous opposing activity? Isn't that just wasted energy?
It seems like it should be, right?
Mm-hmm.
But having a low level of this cycling, where maybe PFK1 and FBPase1 are both slightly active at the same time, actually offers some advantages.
Like what?
Sensitivity and speed. Even in the liver, where regulation is tight, there's probably a small amount, maybe 1–2% of this substrate cycling happening.
Okay.
This slight inefficiency allows the system to respond much more rapidly and sensitively to small changes in regulators. Think of it like having the engine idling allows you to accelerate faster than starting from cold. It amplifies the response to signals.
Ah, okay. So it makes the transitions between states fed, fasted, exercise quicker and more responsive.
Exactly. And in skeletal muscle, this futile cycling between PFK1 and FBPase1 is actually much more significant.
Really? Why in muscle?
It's thought to be crucial for the incredibly rapid surge in glycolysis needed when muscle contraction starts. At rest, PFK1 is more active. But just before contraction, in anticipation, the activity of both enzymes might increase. Then, bam, contraction begins, PFK1 activity skyrockets, FBPase1 activity plummets, and you get that massive, immediate burst of ATP production from glycolysis that muscle work demands. The cycle allows for a much larger fold increase in net glycolytic flux than if you were just turning one enzyme on from zero.
So a controlled inefficiency for the sake of rapid power generation.
Fascinating.
Okay, let's zoom out again. We understand the pathway, the regulation.
Yeah.
How does all this keep our actual blood glucose concentration so stable it stays in a remarkably narrow range?
It really is remarkable. Maintaining that stable blood glucose is critical. In the post-absorptive state, like after sleeping, it's usually around 4.5 to 5.5 millimoles per liter.
Okay.
After a carb-heavy meal, it might bump up to maybe 6.5 to 7.2 for a bit. Even during prolonged starvation, the body fights hard to keep it above, say, 3.3 to 3.9.
And why is that range so critical?
The brain. Your brain relies almost exclusively on glucose for fuel under normal conditions. A sharp, sudden drop in blood glucose, severe hypoglycemia, can cause serious problems like confusion, seizures, even coma.
Right.
Though the body can adapt somewhat if the hypoglycemia develops slowly. And it's interesting, different animals have different normal ranges. Birds run much higher glucose levels. Ruminants, like cows, run significantly lower.
Why lower in ruminants? We touched on this with propionate.
Exactly. Because of all that fermentation in their rumen, they absorb large amounts of short-chain fatty acids, acetate, propionate, butyrate.
Right.
These become their primary fuels, especially after feeding, reducing their immediate reliance on glucose compared to us.
Got it. So for us, where does the glucose in our blood actually come from? We know gluconeogenesis is one source.
Right. There are three main contributors to your blood glucose pool. 1, carbohydrates from your diet.
Obvious one.
2, gluconeogenesis, making new glucose primarily in the liver and kidneys, as we've detailed. And 3, glycogenolysis, breaking down stored glycogen, mainly in the liver.
Stored glucose reserves.
Yep. Dietary carbs get broken down to glucose, galactose, and fructose. They get absorbed, go to the liver via the portal vein, and the liver quickly converts galactose and fructose into glucose or glucose metabolites.
No.
Gluconeogenesis uses those non-carb precursors. And liver glycogenolysis provides a rapid release of stored glucose when the blood levels start to dip between meals or overnight.
You mentioned the Cori cycle and glucose-alanine cycle. How do they fit into this overall picture of maintaining blood glucose?
They're great examples of inter-organ cooperation. The Cori cycle, lactate, produced by muscles during intense activity, or by red blood cells all the time, travels in the blood to the liver and kidneys. There, it's used as a substrate for gluconeogenesis. The liver makes new glucose from the lactate and releases it back into the blood for other tissues to use. It's a recycling system.
Recycling lactate back to glucose.
Exactly. The glucose-alanine cycle is similar, but involves the amino acid alanine. During fasting, muscles might break down some protein. The nitrogen from those amino acids can be transferred to pyruvate, which might come from glucose or glycogen breakdown, to form alanine.
Okay.
Alanine travels to the liver. In the liver, the nitrogen is removed and eventually made into urea, leaving pyruvate again, which can then be used for gluconeogenesis.
So it's a way to get muscle protein carbon to the liver for glucose production and also transport nitrogen safely.
Precisely. It indirectly allows muscle fuel stores to contribute to blood glucose maintenance. And don't forget glycerol from fat breakdown. That's a continuous source of carbon for gluconeogenesis during fasting. And the energy for all this liver gluconeogenesis, that comes mainly from burning fatty acids, also released from adipose tissue.
It really is a whole body coordinated effort. The liver seems absolutely central. How does the liver know what the blood glucose level is and how to respond?
Liver cells have a special glucose transporter called GLUT2. It has a high capacity and, importantly, a low affinity for glucose. It works in both directions.
Low affinity, meaning it only works well when glucose is high.
Not exactly. It means it's not easily saturated. The rate of glucose transport is basically proportional to the blood glucose concentration over a wide physiological range. So the glucose level inside liver cells pretty much mirrors the glucose level in the blood flowing through it.
So the liver gets a direct reading. How does that compare to other tissues?
Most other tissues like muscle and fat rely on transporters like GLUT4. These have a higher affinity but lower capacity, and critically their activity, or rather their presence on the cell surface, is regulated by insulin. Pancreatic beta cells are an exception. They also use GLUT2, which is key for sensing glucose to release insulin.
So the liver is always open to glucose flux, while other tissues are more regulated by insulin gates.
That's a good way to think about it. This difference allows the liver to act as a glucose sensor and buffer for the whole body.
Let's circle back to glucokinase in the liver. You said it has different properties from hexokinase. How does that play into this buffering role?
Right. Hexokinase, found everywhere, including the liver, has a low kinemator for glucose. It gets saturated, works at its maximum rate, even at normal fasting glucose levels. It takes care of the liver's basic glucose needs.
Okay.
Glucokinase, however, is the main player in the liver and pancreas beta cells for handling large glucose loads. It has a high kinemator. This means its activity increases significantly as glucose concentration rises, like in the portal vein, blood coming directly from your gut after a meal.
So it ramps up when there's lots of glucose to deal with.
Exactly. This allows the liver to efficiently trap large amounts of incoming glucose by phosphorylating it, preventing excessive spikes in peripheral blood sugar. It can then channel that glucose 6-phosphate into glycogen storage, or, if stores are full, potentially towards fatty acid synthesis.
And you mentioned it moves location too.
Yes. During fasting, glucokinesis is mostly kept sequestered in the nucleus of liver cells. When glucose levels rise after a meal, it moves out into the cytoplasm where it can phosphorylate incoming glucose. This movement is regulated, adding another layer of control.
Fascinating. So the liver switches from being a glucose producer during fasting to a glucose taker-upper after a meal, thanks to GLUT2 and glucokinase.
Precisely. At normal fasting levels, the liver's putting glucose out. As blood glucose rises post-meal, that output stops, and the liver becomes a net consumer, soaking up glucose. Ruminants, by the way, lack significant glucokinase activity, which fits with their different digestive pattern, no big glucose flood from the gut.
Okay, this intricate system relies heavily on hormones. Let's talk insulin and glucagon. They're the main duo, right?
Absolutely, the main players in moment-to-moment glucose regulation. Insulin is secreted by the beta cells in the pancreas when blood glucose goes up.
How do the beta cells know?
They use GLUT2 to take up glucose and glucokinase to phosphorylate it, just like the liver. This increases ATP production inside the beta cell. That rise in ATP closes special potassium channels on the cell surface.
Okay.
Closing those channels changes the electrical charge across the membrane depolarization. This opens calcium channels, calcium flows in, and that triggers the release of stored insulin granules out of the cell.
Wow, a direct link from glucose level to insulin release.
Yep. So blood insulin levels generally track blood glucose levels pretty closely. Other things can stimulate insulin release too, certain amino acids, fatty acids, ketone bodies, even glucagon itself, some gut hormones, and certain drugs like sulfonylurea is used to treat diabetes. Epinephrine, the stress hormone, actually inhibits insulin release.
And what does insulin do once it's released?
Its immediate big effect is to signal muscle and adipose tissue to put more GLUT4 transporters on their cell surface. This dramatically increases their glucose uptake, lowering blood glucose.
So it opens the gates in muscle and fat.
Exactly. Insulin doesn't directly increase glucose transport into the liver via GLUT2, but it promotes overall glucose utilization in the liver. It encourages glycogen synthesis by activating glycogen synthase and inhibiting glycogen phosphorylase, the breakdown enzyme, and it pushes glycolysis forward, partly through effects related to glucokinase. Longer term, insulin acts at the gene level, promoting the synthesis of enzymes for glucose use and storage, while suppressing the enzymes for glucose production, gluconeogenesis.
Okay, so insulin is the lower blood sugar hormone. Glucagon does the opposite.
Precisely. Glucagon is secreted by the alpha cells of the pancreas when blood glucose gets low. Its main target is the liver.
Just the liver.
Primarily the liver for its major effects. In the liver, glucagon stimulates the breakdown of stored glycogen, glycogenolysis, by activating glycogen phosphorylase. This releases glucose into the blood. Unlike epinephrine, glucagon doesn't really affect muscle glycogen breakdown.
Okay.
Glucagon also strongly promotes gluconeogenesis in the liver, stimulating the enzymes involved in making new glucose from amino acids and lactate.
How does it do that?
Mostly via that CAMP pathway we talked about earlier. Glucagon binds its receptor, activates adenylyl cyclase, makes CAMP, activates pKa, and pKa phosphorylates target proteins that lead to increased glycogen breakdown and gluconeogenesis. So glucagon raises blood sugar by telling the liver to release stored glucose and make new glucose. It's the direct counter-regulatory hormone to insulin.
And the liver cleans them both up afterwards.
Yes, the liver plays a major role in clearing both insulin and glucagon from the circulation, helping to terminate their signals. It's really the central command post for glucose.
Are there other hormones involved in this balancing act?
Oh yes. Several others fine-tune things or respond to different situations. Hormones from the anterior pituitary, like growth hormone and ACTH, which controls cortisol release, tend to raise blood glucose and generally oppose insulin's actions.
How does growth hormone do that?
Growth hormone, which is actually stimulated by low blood sugar, seems to decrease glucose uptake by muscles. This might be partly indirect. It promotes fat breakdown, and the resulting increase in fatty acids can interfere with glucose use in muscle.
Okay. And cortisol, glucocorticoids.
Glucocorticoids, like cortisol from the adrenal cortex and some made in fat tissue too, definitely raise blood glucose. They do this mainly by boosting gluconeogenesis in the liver. They increase protein breakdown, providing amino acids, and they induce the synthesis of key gluconeogenic enzymes. They also tend to make peripheral tissues like muscle less sensitive to insulin.
So stress hormones generally raise blood sugar.
Right. And interestingly, in obesity, immune cells called macrophages in fat tissue release inflammatory signals, cytokines, that also contribute to insulin resistance. It's complex. Then there's epinephrine, or adrenaline, from the adrenal medulla.
Yeah.
Released during stress, fight, or flight. Epinephrine rapidly stimulates glycogen breakdown in both the liver, releasing glucose, and muscle, fueling glycolysis for muscle activity, producing lactate. Because it increases lactate and potentially glycerol release, it's also a potent stimulator of gluconeogenesis.
So a whole team of hormones working together, sometimes opposing each other, to keep glucose in that tight range. Let's shift to when things go wrong. Clinical aspects. What happens if this regulation breaks down? You mentioned glucosuria?
Yes, glucosuria, glucose in the urine. This happens when your blood glucose level gets too high, exceeding the kidney's capacity to reabsorb all the glucose that gets filtered out of the blood.
There's a limit to how much the kidneys can pull back.
Exactly. It's called the renal threshold, typically around 10 millimoles per liter, or 180 milligrams per deciliter. Above that, the reabsorption machinery is saturated, and the excess glucose spills into the urine.
And that's common in diabetes.
It's a classic sign of poorly controlled diabetes mellitus. This loss of glucose in the urine contributes to weight loss because you're losing calories. It also pulls water out with it by osmosis, leading to increased urination and dehydration osmotic diuresis.
Makes sense. You also mentioned hypoglycemia risk during pregnancy and in newborns. Why then?
During pregnancy, the growing fetus constantly draws glucose from the mother's blood. It's the baby's main fuel. This extra demand can make the mother more prone to low blood sugar, especially if she goes long periods without eating.
Okay. And newborns?
Newborns, especially if they're premature or small, face several challenges. They have limited fat stores, so they can't rely as much on fatty acids for energy. Their gluconeogenic pathways might not be fully mature yet, and making glucose requires energy, often from fat oxidation. They also don't have large glycerol reserves. All of this makes them vulnerable to hypoglycemia, particularly between feeds.
So careful feeding schedules are important. How do doctors actually check how well someone's glucose regulation system is working?
The classic test is the oral glucose tolerance test, or OGTT. You fast overnight, then drink a standardized sugary drink. Then blood glucose is measured periodically over the next couple of hours.
What does it show?
It shows how quickly and effectively your body clears that glucose load from the blood. This depends on both how much insulin your pancreas releases and how sensitive your tissues are to that insulin. Insulin resistance, often linked to weight gain, might show up as a slower return to normal glucose levels. Initially, the pancreas might compensate by pumping out extra insulin, but eventually that can fail. The OGTT is especially used to screen for gestational diabetes and pregnancy.
Is there a longer-term measure?
Yes, a very important one is HbA1c, or glycated hemoglobin. This measures how much glucose has attached to your hemoglobin molecules in red blood cells over time. Since red blood cells live for about 2-3 months, HbA1c gives you an average picture of blood sugar control over that period.
What are the typical levels?
Below 6.0% is generally normal. 6.0% to 6.4% is considered pre-diabetes range, indicating increased risk. 6.5% or higher on 2 separate occasions is usually diagnostic for diabetes mellitus.
And diabetes itself is essentially a failure of this system.
Fundamentally, yes. Diabetes mellitus is defined by chronic high blood sugar hyperglycemia due to problems with insulin. In type 1 diabetes, the immune system destroys the pancreatic beta cells, so there's little or no insulin secretion. It usually starts in childhood or young adulthood.
Okay, autoimmune.
Right. Type 2 diabetes is much more common, especially in adults, and is linked to lifestyle factors like obesity and inactivity, though genetics plays a role too. It involves 2 core problems: insulin resistance, where tissues don't respond properly to insulin, and eventually impaired insulin secretion, where the beta cells can't keep up with the increased demand.
Insulin resistance seems key in type 2.
It is, and it's often part of a larger picture called the metabolic syndrome, a cluster, including abdominal obesity, high blood pressure, abnormal blood fats, and increased risk for heart disease and stroke, along with the high risk of developing full-blown type 2 diabetes. Impaired glucose tolerance can also show up in other situations like severe liver disease, some infections with certain medications, or if the pituitary or adrenal glands are overactive, producing hormones that counteract insulin.
What happens if you give someone insulin, or too much insulin?
Giving insulin therapeutically lowers blood glucose by boosting uptake and storage. But too much insulin causes hypoglycemia, dangerously low blood sugar. Symptoms can range from sweating and shakiness to confusion, seizures, coma, and it can be fatal if not treated quickly with glucose.
And can hypoglycemia happen without taking insulin?
Yes. Fasting hypoglycemia can occur if the counterregulatory systems aren't working well. For example, in conditions where the pituitary or adrenal glands are producing enough of those insulin-opposing hormones, or if liver disease severely impairs gluconeogenesis, someone might become hypoglycemic after fasting.
Okay. Now, the text includes a section on very low carbohydrate diets. How does gluconeogenesis fit into that discussion?
Right. These diets, often called ketogenic diets, restrict carbs very severely, maybe 20 grams a day or less, while allowing lots of fat and protein. They definitely force the body to rely heavily on gluconeogenesis.
To make the glucose the brain still needs.
Exactly. The body has to make glucose from amino acids, from protein, and glycerol from fat. This process, as we've said, costs energy, ATP, which comes mainly from burning fatty acids. Some proponents suggested this increased gluconeogenesis and fat burning might lead to greater weight loss than just calorie restriction alone, a metabolic advantage.
But is that supported by evidence?
Controlled studies generally haven't found a significant increase in total energy expenditure. The weight loss seen on these diets seems to be primarily due to reduced overall calorie intake. High protein and fat are very satiating, and the restricted food choices often lead people to eat less overall.
So a calorie is still a calorie, basically.
That seems to be the case regarding energy expenditure. There's also often rapid initial weight loss on these diets, but a large part of that is likely water loss. When you deplete your glycogen stores in the liver and muscle, right, glycogen holds water. Exactly, about 3 grams of water per gram of glycogen. So as glycogen goes down, you lose that associated water weight quickly. The real challenge with these diets, like any restrictive diet, is often long-term adherence.
So gluconeogenesis is working overtime on these diets to supply essential glucose, but the weight loss isn't some magic metabolic trick, more likely calorie control and water loss. Understood. Wow, we have really covered a massive amount of detail on gluconeogenesis today. To try and wrap it up, what's the absolute core message about why this pathway is so vital?
I'd say gluconeogenesis is fundamentally about survival and stability. It's your body's non-negotiable mechanism for ensuring glucose availability, particularly for the brain, when dietary sources run low. It's not just a backup, it's a constantly regulated process working in elegant reciprocal partnership with glycolysis. Understanding how it's controlled the enzymes, the allosteric effectors like fructose, 2-prefix-bisphosphate, the hormones like insulin and glucagon is absolutely critical for understanding normal metabolism and also for tackling major health issues like diabetes and hypoglycemia. It's truly a cornerstone of metabolic homeostasis.
Absolutely. And maybe a final thought for you, our listener, to ponder. Given the intricate design and energy cost of gluconeogenesis, what does its evolution tell us about the metabolic tightropes our ancestors walked, needing to guarantee fuel even through periods of scarcity? Perhaps this dive has also made you curious about digging deeper into insulin resistance, the different facets of diabetes, or how various diets really impact these complex metabolic pathways. Thanks for joining us for this deep dive into the essential process of gluconeogenesis.